So this section is on genetics and evolution. I mean, we can't have a course on biopsychology without talking about some of the most fundamental, important parts of biology. Also, genetics and evolution are playing a bigger and bigger role in how we understand behavior, how we understand animal behavior, and how we understand human behavior. So we'll get into that as well. So the fundamental chemical involved in transferring of information from one generation to the next and for coding for how our body works is DNA. I'm sure you've heard of it before, deoxyribonucleic acid. And it's simply a double helix, and it is a recipe book. That's all it is. It's a recipe book of information. Now that recipe book is written with 4 letters. A, adenine, G, guanine, T, thymine, and C, cytosine. These are just letters of the DNA. And this is the language that we use. What's interesting is that most of our DNA we don't even code for anymore. We don't even use anymore. So the part of the DNA that we code for, that's going to be called a gene, and its function is to code for protein, how to make protein. That's really all your DNA does, primarily, is a recipe book for protein. And then protein regulates so many things. It regulates and creates enzymes. It has a lot to do with neurotransmitters, its building blocks. And so that's pretty much what we're going to do. We're going to code from DNA. We're going to take that code in the form of RNA. We're going to take it over to the ribosomes. We're going to make protein. Now, the human genome has 23 pairs of chromosomes. Now, chromosome 1 is the largest, and then you go all the way down to chromosome 21, 22, and 23. And really 23 are sex chromosomes, the X and the Y chromosome. Okay? Now, again, remember that a gene is just a code for protein, and those genes are found on our chromosomes because chromosomes are just wound up DNA. Now, what's also important to know is that oftentimes, but not always, your genes are represented by 2 different forms, or 2 of the same forms. You get one from your mom and one from your dad, and those are called alleles. So let's say you have a gene for hair color. Now you're going to get some information from mom, from dad, and that information, those types of genes are known as alleles. And we're going to come back to alleles in just a bit. Okay, so when we talk about the gene expression, the way the alleles are put together, we talk about a genotype. These are the genes. And you might have 2 forms of the gene. You have maybe a dominant and a recessive, a dominant and a dominant, or 2 recessive. Then the expression of the gene, what the gene actually does, what it looks like, what it creates, what we see, is the phenotype. So the representation of the gene, or what's going to happen, is the phenotype, and the actual genes themselves is known as the genotype. When we look at genotype and we're looking at, let's say, these 2 strands of a chromosome, one that you got from mom, one that you got from dad, you might have, again, 2 dominant alleles. We call that homozygote. Homo meaning same. Homozygote dominant. You might have homozygote recessive, 2 recessive alleles. But you might have one of each. And this is known as hetero, or different. Heterozygote. There are a couple types of phenotypes that are kind of interesting and easy to see because they are created from only 1 gene or maybe 2 genes. Okay? So, for example, rolling your tongue. Can you roll your tongue like this person here? Give it a try. If so, you have at least one of the dominant genes. You're either heterozygote or you're homozygote dominant. If you can't roll your tongue, then we know you have 2 recessive alleles for tongue rolling. Okay? Eye color is really the combination of a couple of genes. Attached earlobes, like this, that is a product of a dominant allele. If you have one of them, either heterozygote Or you are homozygote dominant, you have a big floppy earlobe. But some people have attached earlobes. Those are homozygote recessive. There's a whole bunch of these types of monozygotic traits. Well, let's look at a couple of kind of strange examples, because oftentimes diseases or disorders are the result of homozygote recessive, two recessive alleles. And if somebody's heterozygote, they have a dominant and a recessive, they don't express the disorder. And there's a lot of really bad ones. Tay-Sachs, sickle cell anemia like this, cystic fibrosis. And the question is if some of these disorders are very deadly, like sickle cell can be, how can the recessive allele keep going through a population? Well, one, it can be in the heterozygote form. They have a dominant allele, they have a recessive allele, the recessive allele is there, but they don't express it. They aren't sickle cell. Sickle cell anemia, let's take a look at that. These are blood cells that are round. Normally, your blood cells are round, but somebody with this disorder has these sickle-shaped blood vessels. These sickle-shaped blood cells, and they don't go through blood vessels very easily, like capillaries and things, and they can get stuck, and it can be very, very painful. So somebody who's homozygote dominant has round blood cells, and they're fine. Somebody who's heterozygote, they have one of each type of allele, they're fine. But somebody with two recessive alleles, well, that can be very bad and very deadly. But it turns out that in this case, and many other cases, a heterozygote form is actually the best form to be, because somebody who's heterozygote might be resistant to malaria. So here is this sort of coevolution. You get malaria from getting bit by a certain type of mosquito, and it affects your blood cells, and it can be very deadly. But they don't tend to infect the blood cells of somebody who's a carrier, who's heterozygote. Now, somebody who's heterozygote has round blood cells, they're fine, but they're also resistant to malaria. So they survive better than somebody who's homozygote dominant, who has two dominant alleles. Now, their blood cells are fine, but they also can get malaria. So let's think about this. Let's say there's a female, and she has two types of eggs. Because while you have 23 pairs of chromosomes in all of your cells except your sex cells, like sperm and eggs, they only have 23. We got to split them up so that when they reunite, you're back to 23 pairs. So when you split them up, each type of allele can go into each type of sex cell, like eggs. So let's take this female here. She is heterozygote. I mean, she's a carrier for sickle cell. And let's say a man, his sperm he's also heterozygote. This is called a Punnett square. And we can think about probabilities of the egg and sperm getting together. So let's say this egg and this sperm get together, and you end up with somebody who is homozygote dominant. Their cells, their blood cells, are just fine. But they also be very susceptible to malaria. And by the way, even today, malaria kills about 3 million people a year, primarily in areas that are near the equator. But for a long time, it killed a lot more people than that. So this individual right here would be a dominant allele in the sperm and a recessive allele in the egg, and they're heterozygote. Same with this person here. This is a recessive allele from the sperm and a dominant from the female. And then that person is homozygote recessive. So let's say two people get together and they are both heterozygote for sickle cell. What's the probability of their child having actually the sickle cell anemia, the problem with the blood cells? Well, there are 4 possibilities. One of them is homozygote recessive, so it's a 25% chance. So let's think about this individual. Here's somebody whose blood cells are fine, but if they live in an area with high malaria, then they get malaria and die. So that's not the best what we call an evolutionary stable strategy. In other words, it just doesn't work well if they live near the equator. This person down here has sickle cell, and they die young, so they don't pass on their genes. This is before modern medicine. So it's really the heterozygote that is the one that is really the best to have. We see this with a lot of different types of disorders. For example, Tay-Sachs. Somebody with Tay-Sachs is a terrible disease. It's a homozygote recessive disease, and they die. But if they're heterozygote, they don't get Tay-Sachs disease, but they also are more resistant to tuberculosis. So now heterozygote is a good way to be, but two heterozygote people getting together, and they have a 25% chance of having somebody with Tay-Sachs disease. So I wanted to talk a little bit just about some interesting things that are happening in genomics, in the field of genetics and biotechnology. One of them is that we can take a gene. Well, we take genes from just about, a lot of different animals and put them in different animals. We can take genes from animals and put them in plants. We can put them from plants to other plants. This is known as genetic modification. And most of the food you eat in the United States, corn and soy and all, these are all genetically modified. These are organisms that have the genes from other organisms, because we all have the same language. An insect's DNA, a corn's DNA, a flower's DNA, right? Some type of flower, and my DNA, we're all the same. We just put our DNA in slightly different order. Well, what's kind of interesting is one, we can take a green fluorescent protein, and actually we can change these to a lot of different colors, that comes from a bioluminescent jellyfish. This animal glows in the dark. And we can remove that, we can find that gene, we can isolate that gene, we can take it out and just put it into another animal. So we have these glow-in-the-dark mice, we have glow-in-the-dark pig, we have glow-in-the-dark fish. You can actually buy glow-in-the-dark fish, genetically modified fish, in Texas. You can't in California, though. And so we're doing a lot of this now. Now this green fluorescent protein is being used in tremendous ways to identify different parts of the brain, different types of cells, different cancer cells. So there is this one type of mouse, genetically modified mouse, called a rainbow. Rainbow because its brain is a rainbow of different colors, of different fluorescent proteins added to different parts of the nervous system to make these cells glow in the dark. We've really gone a long way in terms of tracing different pathways in the brain because of this. This is all through genetic modification. I think that we are moving forward very quickly in terms of this genetic modification to do a lot of things. For example, there is work to clone pigs or to genetically modify pigs so that they have similar sugars and proteins on their muscles, like their heart muscle, that is similar to humans. So we could take the heart out of this genetically modified pig and implant it into a human. And since people who are on transplant lists, sometimes they die on these lists because there's not enough organs to go around, this might be a way of doing this. We're also doing this thing called 3D printing, which is 3D printing of genetically modified tissue. We're printing organs. One of my favorite examples is the spider goat. I love the spider goat because it makes me laugh just thinking about it. Here's a goat that's been genetically modified. We took the DNA sequence that codes for certain types of proteins that are in spider webs. We took the genes from spiders and we put them in a goat. And now the goat produces milk that has lots and lots of these proteins that are then spun and turned into these ultra heavy duty fibers. And they're thinking about making bulletproof clothing out of these fibers, or having these fibers that have the strength of steel but they're the size of threads. this is some strange stuff going on. Well, another thing that my wife and I and our kids did over the summer is we went to a website called 23andMe. And they sent us a package, and we spit into these little containers, and we sent off our DNA. And we got the DNA back from this company. We can go online, we can look at a lot of our protein markers, a lot of our DNA markers for susceptibility to different diseases, or how we relate to different types of drugs, and whether we're at a risk for high cholesterol or heart disease or all these different kinds of things, including Parkinson's and Alzheimer's. We even got back our ancestral history of where we came from, and who we're related to. And we get emails all the time about a third cousin or a second cousin somewhere in the United States or even around the world that we're related to. This is a lot of information because DNA sequencing has become so incredibly cheap. Used to be terribly expensive, prohibitively expensive, and now you can get your whole DNA sequence for about $80. This is some interesting information. I found out something that was really surprising to me. I'm in the 90th percentile for Neanderthal. I have Neanderthal DNA greater than 90% of the population, and my son 92%. So we're Neanderthals. So I want to just give a little bit of a background about evolution. I think as many classes as you can have to get some basics of evolution, because there's a lot of misconceptions about evolution out there, but also these ideas of genetics and evolution and heritability are very important and are becoming more important in the field of psychology. And I think if you decide to go on and work in applied aspects of psychology and therapy, I think you're going to start using the notion of genetics as influencing not only maybe prescriptions of psychoactive drugs, but therapy techniques. So let's talk a little bit about the basics of evolution and the theory of natural selection. I always like this quote: "Nothing in biology makes sense except in the light of evolution." It's our foundation of medicine. It's our foundation of genetics. It's our foundation of so many things to understand this basic process. So Charles Darwin, interesting story of him. I'm just going to give a little brief history. Here's a very smart person and a bit of an aristocratic family. And he tried to go into medicine and really didn't have the stomach for it, didn't like all the blood and guts and screaming. By the way, they didn't have anesthesia when he went through medical school. But he loved nature. He really loved nature, and he loved being outdoors. And he got the chance to go on a journey for 5 years, not really as the naturalist of a ship. The ship was called the Beagle, but really as almost like a companion to the captain, who was named Fitzroy, because they were both from these aristocratic families. But the thing is that Charles Darwin did not like being on a ship. He threw up all the time, very sick. So every time the ship landed, this exploration, the Beagle, he got off and stayed on land as long as he could. In fact, there were times where he stayed weeks on end, and they had to go and get him before the ship left. And he collected and he collected and he collected, and he wrote great notes, and he sent back all of his collections back to Britain on ships going in that way. And here's the famous Galapagos Islands over here, and wrote down all these great observations about nature. And he read books about geology and the possible really old age of the Earth. And he read books about math and about population dynamics and all these things, and started putting it together because people had thought about animals and plants changing over time, but nobody really had a really good theory about it. So I want to talk about his theory. It's a theory called natural selection, or modification by natural selection, of the process of evolution. There's not a theory of evolution, there's a theory of the mechanisms of evolving organisms. And I think it's important just to lay out the very fundamentals of his ideas. First thing that he noticed, and others have noticed, is that all organisms tend to overproduce. They produce more offspring than the environment can handle. Right? So mice make, they have little mice pups, and there's 10 of them, and then they have 10 more, and then 10 more. And if all the mice pups that this two pair of mice have, if they all survive to reproduce, we would be knee deep in mice in a matter of months. So he noticed that all organisms, plants and animals and fungi and everything, over reproduce, and yet populations tend to stay stable. What does that tell you? It tells you that not all the organisms born or hatched or planted can grow to be sexually mature to reproduce themselves. So, number one, all organisms tend to over reproduce. The second one that he noticed, and he wrote a lot about this, is the nature of variation, of variability, of variation in phenotypes. There are differences. There's differences everywhere. Look at the people around you. Look at your friends. You have different skin tones, skin color, hair. Some of you get colds more easily. Some have good lungs for running. Some can sprint. Some can go distances. Where there's just so many differences. We are a varied population of animals. But that's true for all organisms. There is variation within a population. Now, a population, by definition, is interbreeding species. So if you have a population of squirrels, these squirrels have the possibility of interbreeding because they live near each other, they breed at the same time, but there's variation. There's variation in how they look and how they run and how they smell and all those kinds of things. So number one, over reproduction. Number two, all organisms show a variation. The third one is the idea that there is competition. If all organisms over reproduce, who gets to survive to reproduce? Who gets to do it? It's not random. If you ever hear somebody say the theory of natural selection is all about randomness, it's all random, that's not true. There are components to how genes hook together and mutation that has some random components, but it is a selective process. It is not a random process. There is competition. Competition for resources, competition for food and space and mates and all these things. They fight each other, they create these beautiful nests, whatever. Who breeds is not a random coin toss. It is a competition. Competition with nature, competition within a population, competition against nature, and that is the third postulate. That those that tend to reproduce, there's a competition for who gets to do that. So we see wonderful examples of competition at the individual level. Animals fighting each other in the same species. Look at the peacock. I mean, this is a giant, delicious bird that gets chased down and eaten, and he has these giant feathers. But if it helps him reproduce, if it helps him pass on his genes, then that's going to work for him, and that is going to be that's going to be a variation that is selected for. Nest building, there's variation. So imagine a group of animals, and suddenly the climate gets hot for hundreds of years. Well, animals maybe with a little less fat because of variation, a little thicker, a little less thick a wool or fur, they're going to survive better. What if it suddenly gets cold? Then the ones that are warmer are going to survive. There's something for nature to select from. What about sexual selection in humans? There's variation in that as well. These are some very strange, what appear to be strange, but these are selective characteristics, sexual characteristics. And what we see as odd in one culture, others see as odd in our culture. I mean, that is a torturous shoe there. There's even, this guy got calf implants. I found that on the web. Who gets calf implants? Come on. Is that so the guy looks good walking away? I don't get it. But, these are the things. These are the things that we do. And this can change from generation to generation and cultures to cultures. This is the way I think sexual selection should work, but that's just a personal bias. All right. So, there's a tendency for overpopulation. There is variation in all sorts of phenotypes. There is competition for who gets to reproduce. And those, number 4, those that happen to be able to compete better, more successfully, find food better, run better, hide better, survive disease, survive cold, whatever the environment places upon them, whatever members of that population that at that time compete better, on average, tend to reproduce more, tend to survive better, tend to reproduce more, and they pass on those genes that they had that helped them at that time. Nature selects. This is not a random process. This is a selective process. And number 5, we can get modification of organisms from generation to generation. This isn't the idea that a bodybuilder gets big muscles and his baby is born with big muscles. No, that's not true. What it is that the characteristics that they were born with, if the characteristics they were born with, a resistance to disease, a brighter feather, faster, slower, if that characteristics helped them survive that they were born with, then that characteristic in the form of genes will be passed on, and the next generation will have those genes. And we get what's called a modification through descent. Now, where you get big changes is because the world has different pressures. There's different pressures placed on a population over the ages. Colder, hotter, wetter, drier, things change. If there is no differences in pressures, we don't see a big difference in a population's gene pool. We don't see a lot of difference. There has to be pressures, and pressures that change. It's all about what the population is experiencing. If suddenly an animal ends up, a group of animals ends up on an island, and there's nothing to eat them, and there's no competition, and they evolve and they can change really rapidly over the course of generations, not over their lifetime, to accommodate the new environment. So, our Earth has changed dramatically. We have weather systems that blow things in and move things out, and volcanoes, and all sorts of just weather changes, environmental changes that change the pressure of the animals. I mean, think about what humans go around killing a lot of things. That's putting different pressures on the different organisms. And we can see some organisms doing well and some dying off because of the pressures of humans. What about the pressures of climate change? The Earth is heating up. How does that affect migration? How does that affect sexual reproduction? All those kinds of things. We see changes. If the world changes, organisms change too. If there's no change in an environment, then we get what's called stasis, which is very little change. But then change can happen rather rapidly. And that's one of the sort of a slight modification to Darwin's theory. Darwin thought of more of a gradual change, and we think today we see things sort of change rather rapidly over the course of thousands of years, as opposed to millions, and then stasis. It's called punctuated equilibrium. Punctuated equilibrium. Equilibrium, very little change, punctuated by rapid change. Let me give you one example. There's a bunch of dinosaurs hanging out 65 million years ago. Dinosaurs are eating everything. Tiny little furry mammals are barely getting by. A comet hits off the Yucatan Peninsula in Mexico, and suddenly all the dinosaurs are dying off. Well, opportunity for the mammals. And suddenly there's not the predation, there's not the competition for food and resources, and there's a tremendous explosion of change in mammals at that time. It's all about opportunity. So we have different pressures that are not only environmental, but we have behavioral pressures as well. Hunting and mating and migration and tool use and all these things. These are subjected to natural selection pressures just like anything else. Behavioral changes. So what's some of the evidence? And by the way, to really fully get a good understanding of natural selection and evolution, there's some really great books out there. It takes study, it takes reading. But I'm just sort of giving you the 10-cent tour. One of the evidence is the fact of we see homologies. These are similarities. For example, we have homology of structure. These are a bat, a human, lion, horse, frog, whales. These are all mammals. We all have the same bones here. Even a flipper. It has the phalanges, it has the radius, the ulna. It has the same bones. These homologous bones tell us that they probably came from this common ancestor a long time ago. Another evidence that he wrote about was this idea of ontogeny, that ontogeny is our own personal development. There's a very famous saying in biology that ontogeny recapitulates phylogeny. Yeah, that's kind of crazy, I know. But phylogeny is our ancestral history. Ontogeny is our personal history. And the fact is that when we're developing as humans, you know, we kind of have these modified, funky kind of gill slits and these tails, and we kind of, I'm going to say this with a sort of grain of salt, but we kind of evolve from sort of fish-like to human-like during our own embryological development. Look at the similarities in embryos to animals that end up becoming rabbits and chickens and tortoises. So embryologically, we have a lot of similarities. One thing that Darwin wrote a lot about in his book on Origins of the Species is his work on South America and the Galapagos Islands, and he looked at what's called biogeography, and that's the distribution of different animals in different locations, and how he looked at, for example, the very famous example of the finches, the Darwinian finches here, and how they had evolved to fill their niche, their habitat of where they live and how they eat, and how the changes, the modification of these birds had a lot to do with where they were on these different islands. So how animals relate to their geography has a lot to do with giving good evidence that these animals have changed. You know, one of the things that he talked about at the beginning of his book on Origin of the Species, very famous book, is he said we've already done this. We've already modified the heck out of organisms by selective breeding. We call this artificial selection as opposed to natural selection. Instead of nature choosing through competitions, humans come in and go, I like a smaller dog, so I have a bunch of dogs, I'm going to breed the smaller ones. I'm going to only let the smaller ones breed, kill the other ones. And over the course of really a short amount of time, we get small dogs. We get the small Shih Tzus and the Chihuahuas and stuff, or maybe a big dog, or maybe these features. We have changed so many organisms, and we have evidence that we can do this. So we have changed chickens and corn and cats and flowers, all through a selective breeding process. And what we get, and this is an arbitrary term in some sense, in biology we call species. Species are animals that can and typically do interbreed. Species are animals that can and typically do interbreed, but it kind of breaks down sometimes. I mean, there are animals that, like dogs and wolves, which are different species, or coyotes that are different species, as we identify them as biologists, but they still can interbreed and produce puppies. There's even a dolphin and a whale in Hawaii that were in the same enclosure at what's called Sea Life Park, and they bred, and they had a wolfen. Oh, that's crazy. And there's ligers and all those kinds of things. Now, these two animals on the screen here are actually the same species, Canis familiaris. That's the two name of the family dog. But can these two animals interbreed? Probably not, especially if that one's the male. Sorry for that mental image. Okay, so why is this important? I mean, there's a lot of reasons why this is important, but I'm going to go over just a few. The importance of understanding genes and evolution and natural selection and pressures. Well, one of the most important things is that your brain, our brain, really hasn't changed much in certain areas. The neocortex has changed, maybe tripled or quadrupled its size since Australopithecines, maybe 3 million, 4 million years ago. But the part of the brain that deals with hunger and food and maybe even some emotions, they haven't changed a lot. The hypothalamus, you know. And so our brain craves food that was rare 3 million years ago, 5 million years ago, 10 million years ago. What's rare in nature? Well, salts can be rare. Simple sugars can be rare. Fats can be rare. And we need those things. We need salts and sugars and fats to survive. And so we have such a taste for them. Our natural diet didn't have a lot of sugars, these types of simple sugars. And fats, maybe you killed something and ate its fat. Well, more power to you. But today they're everywhere. The sugars and the fats and the salts. And our brain says, get it, get more of it. I want it. It's so delicious. And we eat so much, hundreds of times more sugar than our natural ancestors did. And the fats and our brain doesn't have a shut-off valve at sometimes where we're like, boy, supersize me. Give me a big gulp. Give me the big giant cheeseburger. That's what your old brain is telling you. But you're in a modern society. Evolution tells you to shove that big giant cheeseburger down your gullet. But our ancestors say you should only eat meats a little. We should, eat meats a little, eat sugars a little, because that's our natural diet. So we have a big problem of obesity in our society. It's tripled in 20 years, tripled the levels of obesity. These are issues because we're battling our nature. I want to talk about one other thing that's kind of new. It's kind of new in the field of genetics. It's been around for a little bit, but they're really doing a lot of interesting work with it. This field is epigenetics. This is the idea that our genes don't change. The genes you're born with are the genes you have. Identical twins have the same genes. However, environment, behavior, thoughts, food, these are all things that can affect the expression of those genes. Epigenetics, above the genes. So while our genes are the same, how they're expressed, how the proteins are expressed, and in what quantity and quality has a lot to do with our experiences. So genetics aren't everything. It's our our experience of life. And then we get these what are called epigenetic tags, these things that go onto our DNA that influence the expression of these genes. And the question that we're trying to get at, I think, is, do we pass on those tags? Do we pass on the epigenetics in some fashion? And there's some good evidence that the experience one has in their own lifetime, let's say they go through a starvation or something like that, can really affect things like diabetes and food intake and obesity of grandchildren. It's kind of an interesting new field because it really, really relates and correlates to behavior. One thing I want to leave you with is sort of a cautionary tale about genetics and behavior and intelligence, because we're learning a lot about it. We modify genes. We can modify genes in a lot of organisms. Maybe we'll start doing it in humans. We can test for genes. We can have genes that are markers for this and that disease, but also maybe for aggressive personality or addictive personality or criminality. And we've done a really lousy job in our history of dealing with that assumption. So after Darwin's theories came out, some people twisted them. Twisted his ideas. This is not Darwinian. This is a misunderstanding of Darwinian stuff. But there was a movement in the late 1800s and in the 1900s, and it really kept going, called Eugenics. And this was the idea of promoting good births, promoting good people breeding, because they said, oh, we should pass on good genes, right? Well, Darwin, that's not really how evolution works. It's, it's a competition. There's natural selection. What is good today is not good tomorrow. And the organisms go through changes, modifying in relationship to pressures. But people started thinking about it and saying, well, there's probably good genes and bad genes. And if we want a good society, we should promote good genes. So this isn't, by the way, kind of behind closed doors. You could get degrees in eugenics. I mean, there's some very popular people. And it started with this notion of promoting people with good genes to breed and sterilizing those with the bad genes. Now, you ask your question, who has good genes? And of course, the people who have good genes are always the people that are the ones making the rules. They look at themselves and they say, I'm the rule maker, therefore I must have the good genes. And those people over there and that other country and wherever, they have inferior genes. So it's completely arbitrary and completely stupid. But you can promote, good genes, people with good genes, quote unquote, breeding. They had contests at fair for prettiest children, blah, blah, blah. But then you get into negative eugenics, which is preventing people from having children because you made a decision about their genes. And that can lead to some really dangerous things. So here's a history. This is an American psychologist. This is our field here. The idea was he wanted to test people to find out who had good genes by giving them IQ tests. So a lot of the IQ tests, the Stanford Binet and all those kinds of things that came out as this idea to check for intelligence, and we want the intelligent people to breed. Again, a complete misunderstanding of Darwin. They actually call it social Darwinism, but it's a bastardization of Darwin. And so he started putting together these rules. And he gave people tests that were coming in to the U.S., and he established this testing, and what he found was that people from other countries were feeble-minded and shouldn't be allowed in the United States. 83% of Jews, 80% of Hungarians, 79% of Italians, 87% of Russians were feeble-minded. Don't get me started about people from Asian descent. Again, complete and utter bollocks, as we say in England. It makes no sense, but it did lead to the Immigration Act of 1924, where we prevented people from Eastern Europe, the Jews and the Russians, and people from Italy and people from Asia and India. We prevented them from coming to the country because they would muddy up our gene pool. Some pretty sad times, but it was big. It was huge. And of course, in 1924, who's coming out in 1920, late 20s, 30s? Who's starting to make his mark? This is Germany at the time saying, I see what America's doing, and we should promote good genes, the Aryan race, and I think we should take the next step. So we're going to sterilize people. So the Germans, like the U.S. was doing, but the Germans took it to a different level, began sterilizing all these people, mentally disabled, all those kinds of things. And then these people are consuming things that we need, so they began killing people. Killed the homosexuals, killed the Jews, and killed a lot of people with this misguided notion of purifying genetics and purifying genes. It's a really sad tale that really started in the U.S. and had its growth in the U.S. and England. And so, what I'm trying to say is we're knowing a lot more about genes, and you still see these eugenic ideas creeping up. The idea of modifying for good births and good this and good that. It could be a scary time. So I hope that gave you the foundation of evolution and genetics. And see you next time.